Good morning, everyone. Welcome to the second day of the conference, Silent Transformations. This session will be on how evolution learns. Before we hear about evolution, let's have a look at fashion. So people tend to have different hairstyle. They want to express themselves. But even before people started to experiment with hairstyle, nature started to experiment it on ducks. Yes. The main thing is evolutionary traits may appear to be random, but are they? So this will be the core subject of today's session. We will get an insight into how evolution learns and adapts. Maybe we mistook evolution's sense of fashion as evolution's sense of fashion. Our speaker today is one of the authors of the famous book on evolution, The Origins of Life, Dr. Eörs Szathmary. He is a professor at Department of Plant Taxonomy and Ecology at Eötvös, Loránd University. His current focus is on learning theory of evolution and information transfer in evolution. Today, he will help us understand how intelligent evolution is and its ability to learn. Actually, he is in middle of his sleep, so he is going to give his talk. He will be sitting and delivering his talk. Let's welcome Dr. Eörs on stage.
We discussed it yesterday when Greg was giving his lecture that although evolution in toto may not be algorithmic, but certainly it has got very important algorithmic components, and it's actually rather surprising that one can summarize the essence of Darwinian theory on a single slide. Now, this is quite remarkable and not necessarily welcome, for example, by people in the humanities, because quite a few of them, and I was in an institute in the Collegium Budapest where we were regularly mixed together, in many cases I had the impression that many of them prefer complicated explanations for complex phenomena, right? And if it's not complicated, then it somehow doesn't feel right. Now, it's amazing that in contrast in the natural sciences, you can, at least the principles you can summarize on a single slide. And Darwin was actually aware of these roughly 20 years before publishing The Origin of Species. He was just simmering, and then triggered by the menace that Wallace appeared on stage, he started to write his book, and he referred to The Origin of Species as my wretched book, because it was written in haste and he knew that it wasn't written well. So these are the interesting historical things. But it's true that if you have the case that you have got different kinds of units, A and B and others, and all of them can multiply and show heredity, which means that A gives rise to A, B to B, and variability, which means that occasionally something new appears which wasn't there. And if it's true that some of the hereditary traits affect survival or fertility, the combination of the two we call fitness, then in a population of such units, evolution by natural selection might take place. There are also quantitative conditions. For example, depending on parameter values, the population size cannot be too small because it will be then dominated by random drift, or the mutation rate cannot be very high because then you will go beyond the eigen threshold, so the genetic information will melt. But kurz und gut, as the Germans would say, this gives you a good verbal summary of what kinds of things you can have. This is, by the way, John Maynard Smith. This is his formulation of the characteristics of the units of evolution. So, in fact, it's very simple to begin with. But of course, it's a little bit like Newtonian mechanics. If you want to apply it to a complicated machinery, right, then all of a sudden it blows up and you can very easily calculate for 50 pages. And that's, of course, also true for evolutionary theory if you go into the details. So What I also would like to state, which is very important for our present topic, is about the nature of the variation that is being produced. So partly because of the neo-Darwinian mantra, the idea is that the variation that is being produced is random, but of course one has to understand it in what sense it is random. It is not random in the sense of a uniform distribution, but the idea is that it's random relative to the direction where you want to go. It's random relative to the direction of selection. As we shall see, this is not true any longer, depending on the situation, which is a great thing to learn. So evolutionary theory itself evolves, and some of you might know that there was even a meeting in the Konrad Lorenz Institute a few years ago, out of which a book came out with the title Evolution, the Extended Synthesis. And indeed, I think that their evolutionary theory is in need of some important extensions. Now, the major part of the talk is going to be concerned with topic, how can evolution learn? And this is Dr. Watson, yeah? But for this particular topic, he is actually Sherlock Holmes, and I am Dr. Watson, right? So, but, and if we get to that stage, and I'm still alive, then towards the end of the one-third of the talk, I will probably touch upon a topic where I am Sherlock Holmes, and okay, you see what I mean. Now, this is actually very important because he's an extremely bright lad, and I got to the age where I realized that actually to help talented young people is perhaps more important than helping yourself. So I am kind of a publicity agent for what he realized. I also worked on the subject, but the main idea that you will see what it is actually coming from him. So we should praise him for the insight. Now, the ideas that are going to be expressed have, as usual, certain roots. As people say, a true historian of science is happiest when he or she can demonstrate that something is not original, right? Now, for example, Gregory Bateson had these fantasies. That's probably polite enough, that evolution is similar to a cognitive process. But of course, there was no model or anything to support that claim. We shall see that in a certain sense he was right, but he didn't know why. Before we get to what Richard suggests, let us consider, however, another link which was suggested before. First of all, look at these two quotes. One of them is, inductive inference is the only process known to us by which essentially new knowledge comes into the world. Sir Ronald Fisher. Okay? Richard Dawkins in The Blind Watchmaker. The theory of evolution by cumulative natural selection is the only theory we know of that is in principle capable of explaining the existence of organized complexity. Question, are these claims two different claims, or are they just one claim? There are hints that probably they are just one claim. Here it is. This is something that I'm extremely angry about not to have discovered. You know, because I could have, but shit happens, you know? So I didn't. It was Cosma Chalizi and another guy called Harper, not actually a long time ago. You see, that's a mistake, obviously, that 2010. So he actually, and also Cosma Chalizi, realized that there is an intimate relationship between the discrete time replicator equation and the basic equations of Bayesian inference. Here is the translation table. Here is the population state for the replicator activity. That's the prior distribution of the hypothesis. You have the new evidence here, and the evidence is coming from the environment in forms of the fitness landscape. Here you have got the normalization. Here you have got the mean fitness. And then, of course, you have the updated population state. And then here you have the posterior distribution of the hypothesis. It makes a lot of sense. Any new replicator that is being produced and tested is a hypothesis about how to make a living in the particular environment. It's not a miracle in retrospect that such a relationship exists. And that is one of the deep connections between the two theories. By the way, if you analyze the situation, and I don't want to do it because it's very mathematical, the mathematical relationship between the two is actually very intimate if you are looking at convergences. And so for mathematicians, you know, the Kullback-Leibler divergence plays a very important role. And so the isomorphism is more than superficial between the two systems. So I think that is an insight that we should value. Now, going into the direction of Richard, I would like to have a reminder about something in neuroscience. In neuroscience, one of the important things is that synapses can change because of experience. And one of the canonical forms works in the way that Donald Hebb predicted it. That means that, to put it in a very simple way, that neurons that fire together wire together, which means that if there are two neurons that are connected and they fire roughly at about the same time, then there will be a synapse that is going to be strengthened between them. This doesn't have to do with the direction, which means not the direction, but the order of the firing of the two neurons. That's another form of plasticity that also exists. But the simplest Hebbian rule is just concerned with the rates, which means that the update of the synaptic strengths is going to be proportional to the learning rate and the activity rates of the pre- and the post-synaptic neuron. It's a very simple thing, and it's excellent for detecting correlations. Now, this is very important. So this detects correlations in the environment, not causation, only correlations. But correlations is very good. Actually, it's isomorphic, the principal component analysis in a network, which is quite cool. And we are going to use that. Now, one of the ways where how this can be implemented in a network comes from Hopfield, who was a very famous physicist. And there are various ways of putting it. So here you have got different neurons, and each neuron has got an input and an output. The point is that this is recurrent, so the neurons are connecting to a number of other neurons. In the original Hopfield network, the connectivity was full and also symmetric, which means that the weight from neuron A to B was equal to the weight vice versa. This needn't be so in real situations, but if you make this assumption, you can define a potential function on the system, and you can do all the gimmicks of theoretical physics. And that was the reason for setting it up so nicely. But it is also very important that such a network has very important properties. It's only Hebbian rule. This is what is amazing about it. So imagine that you have got two patterns, not very dissimilar, but not identical either. This is an aeroplane, this is a bird. And then you train the network on the impulses. Of course, you assume that somehow the visual imagery can be directly put into the network or indirectly or whatever, and then it shows very interesting types of behavior. So if you have a random input, then under random input, you will have either this pattern evoked in the system after training, or the other one. Depends, in which direction the randomness goes, so to speak. Much more interesting is that if you have a partial input, it's going to have pattern completion. This is already very important, okay? So a part of the input can trigger what was learned, can trigger the whole encoded information. It can also restore corrupted input, which means that it is blurred by noise. It is a very important thing. So therefore, of course, Hopfield type networks are excellent for memory. Okay, these are different memory traces that you can learn. And it's also true that it can classify. So for example, if you have an ambiguous input here, then this ambiguous input is going to, because it's more similar to an aeroplane than a bird, is going to be classified as an aeroplane. So the aeroplane image is going to be evoked, and the other one is going to be suppressed. So these are the kinds of gimmicks that Hebbian learning can do for you in a recurrent neural network. There are various forms in it. There's a huge body of literature. I don't want to go into that. This is quite involved, but I think that's enough. Now, you don't have to read this. What we were talking so far was this one. This was the Hebbian learning. This actually, I don't know why this is distorted. This should be I and J. This is the update, of the Hebbian synapse as a function of the activities of the two neurons. There are other types of learning. For example, supervised learning. In the case of supervised learning, you want to minimize actively an error rate between the current output and the desired output. And an error signal plays a very important role. Now, please note that if the sign of the error signal is identical to the sign of the activity of the neuron in the Hebbian rule, then the two give identical or qualitatively identical results. So, but there are differences. So to reiterate, the Hebbian rule looks for correlations that are frequent, whether they are good or bad, right? With supervised learning, you are only learning and strengthening good correlations. That's an important difference. And there is another variant, I should have had a slide about it, that's reinforcement learning, where you just give a reward to whatever the animal or the human does. Actually, it works extremely well. I can recommend it to you. We had a lecturer who was pacing up and down during his lecture, not unlike yours truly. But we wanted him to stop that. So we agreed that whenever he was in a particular place on the podium, we would pay very good attention. And whenever he moved away, people were starting, after 15 minutes, without a single movement, he was talking from this position, for the rest of the lesson, right? So it doesn't have to be conscious even, but that is reinforcement learning. You reinforce an element of behavior, and the reward, of course, is the attention. Now, reinforcement learning is very similar to natural selection, as Skinner already realized, because you have got a repertoire, and out of the repertoire you reward certain things, and those things that are rewarded get strengthened, the others are going to be suppressed. It's like selecting for an allele. So there is a very important relationship. Now, before we get into the nitty-gritty, still I have to show you something. This is the life cycle of a lambda phage. The lambda phage is like a game of Space Invaders. It is a virus of a bacterium, and it can get into the bacterium, and it can do two kinds of things. Either it goes immediately lytic, which means that the bacterium is eaten up and is killed, or the DNA of the phage is integrated as a prophage into the chromosome of the bacterium, and this division with the integrated chromosome can go on, and occasionally a prophage exists, the bacterial chromosome initiating lytic cycle, and so on. So you can actually traverse this, and this needs decisions, right? This is what is called a genetic regulatory network. I'm showing this to you because, as this gentleman showed, and this is just one example, Vohradsky from the Czech Republic in the journal Biological Chemistry, he demonstrated how the genetic regulatory network of the lambda phage can be treated as a neural network. Okay, this is important. It's a very important relationship between the two. In fact, generally, it can be said that a genetic network can, in principle, calculate anything that a neural network understood like this, okay? It has to have a formal definition, and vice versa. Now, of course, that doesn't mean that the lambda phage is thinking, because it depends on the actual rendering of the network, whether it does something fancy or not. And of course, there is a difference between the rate constants and so on. But this kind of isomorphism is going to be important for the rest of our talk. A footnote, that's also true for chemical networks. So you could compute anything that you can compute with a neural network with an appropriate chemical network. That has also proven. Has also been proven. But of course, again, there are technical limitations for the realization of a completely chemical computer. Now, the potential domains of applications of the idea that I'm going to discuss with all this introductory material are threefold. You can look at the Evo-Devo, the link between evolution and development, evolution and ecology, and Evo-Ego, as they called it, which is the appearance of higher level organizations in evolution. But that I'm not going to discuss here, because that's the most speculative part, and we are not going to have time for it anyhow. So this was the first paper that I regard as the pioneering paper, The Evolution of Phenotypic Correlations and Developmental Memory. It's Richard, and also immediately the second author is Günther Wagner from Yale, who is an eminent mathematical biologist and developmental geneticist, working both in theory as well as in experiments. So they ganged up, and they defined a model, the process of development. Now, it's, of course, extremely simplified. Be careful. What I'm saying in this talk may or may not be generalizable to other interesting cases. I don't know the limits at the moment, but the promise is so great and so interesting that I think this is why I'm giving this talk rather than something else, which is, so it's terribly old-fashioned because it's real research, right? So I don't know where it will go and so on, but it's terribly exciting. So what we are going to do is that we are going to define the phenotype of this particular model organism with a vector of the activity levels of the different genes it has. And for that, you need kind of an update rule. This is, of course, when it is 0, that's the beginning of the development of the organism. Pi0, the activity levels of the different genes at time 0, is going to be equal to the direct genetic influence that the gene itself has on its own activity, right? This is always how we start, and that's inherited. But there will be another part which will be even more important, how the different genes are acting on each other. So the next generation, this is discrete time, okay, so next time step level of this activity is the previous one. Then there is a spontaneous decay, because molecules are decaying. And here you have got an important, this is something which will be similar to a learning rate. And this is a sigmoid function. And in this sigmoid function, you have the sum of the activities of all the genes. And these are the constants whereby the activity of gene J is influencing the activity of gene I. So this beta matrix is going to be very important, because this specifies the interactions between the genes. And because of the sigmoid nature, it introduces nonlinearity into the model. Now, this is again a vast simplification, because if you think in terms of molecules, this would mean something that these influences are additive, right? You have the influence by one gene and the other gene and so on. But not all genetic regulation works that way, right? Sometimes you have a gene product from gene 1 and gene product from gene 2. And the proteins, the regulators, have to stick together, and only together they are going to act on a third gene. That's not additive, that's multiplicative. But that's not included in this model. But biology is awful, because you have to make these decisions, and you have to start somewhere. It's not like physics, where you have this beautiful phenomena of universality from very simple models. In biology, very simple models are usually not universal, but we have to start somewhere to explore the situation. But it's a caveat that it's not a full model or whatever of development in any way, and there is no space, there are no cells. You assume that basically everybody is in the same pot. Maybe it's a single-cellular organism. Okay, now here is the selective environment. The selective environment is a vector which gives you the desired values of the genes. So this list, this vector is giving you, put it another way, if an organism has this activity level as specified in the vector S, that has the highest fitness. And the fitness of a particular phenotype P star is equal to 1 plus the scalar product between this target vector and the phenotype vector. This specifies the model. That I skipped that. This is going. Okay. So what happens if you are selecting for a single phenotype? Which means that a single phenotype, the single phenotype that you are selecting for is given by this vector S. That is your target. So some of the genes will be upregulated, some of the genes will be downregulated. It goes like this. You start somewhere which is, of course, far from the target that you want. And then you make the first round of development. It's a fixed number of steps. Then you assess the phenotype. Okay? That gives you a fitness. Then you go to the next generation. It's important that the direct genetic effects that I mentioned, that give you the initial condition of the activity level of the genes, changes faster than the B matrix changes. The B matrix is the links between the different genes, and the B matrix changes more slowly than the direct effect on any of the genes. The interesting thing for us is B, the regulation coefficients. So some of them go up, some of them go down. They tend to be evolve in a symmetrical way. It's not quantitatively absolutely symmetrical, but almost symmetrical. The self-interactions are also rather strong. But it's the pattern that is interesting. So here is the interaction between the different genes. This is a matrix, and this gives you the scale. And that's the end result after selection. You can look at the evolved genetic network that produces the best phenotype. What does it look like? What is the B matrix of it? And here is basically the B matrix scaled. Now, the amazing thing is that you can get exactly the same result. You can get exactly the same result if you look at the Hebbian rule between the target activity levels of the corresponding gene. So you remember, in the target phenotype, you have got these S values that can be large or small, but positive and negative also. The Hebb rule gives you a rule, in which direction this should evolve, if you follow the Hebb rule. And that gives also a matrix to you. The amazing thing is that after some scaling, which means that you have an affine transformation or whatever, you can predict from Hebb's rule the evolved matrix of the correlations. Now that's the real insight. So it is a recurrent genetic network that is sort of isomorphic to a recurrent neural network. Your recurrent neural networks use Hebbian rules in order to store correlations that can be recalled. And once you learned, so to speak, by natural selection, how to build the organism with the high fitness, then there will be a certain interaction pattern between the genes. And the interaction patterns, you will find that you could have predicted them from the product values of the pairs of genes of the target phenotype vector. This is the amazing thing. And you can have all kinds of other investigations that I don't want to go into it. But this is only one selective environment. If you do the experiment in the following way, that you select for a particular phenotype for 2,000 generations, then you switch to another phenotype for another 2,000 generations, then you will get something very interesting. After this training, or after this natural selection for two target patterns, taking turns, once you are through, you will find that now you have got three types of activities of gene levels. So some of the gene activities go up, others go down, and some stay very close to zero. When you look now at the interaction matrix that you evolve, that's again a B matrix. But the B matrix is now under the constraint of a double selection, right? So the question is, how do they relate to each other? You see, it's a much more dodgy or interesting pattern, because as you can expect, there are also contradictions between the two environments and so on. But again, if you look at the different values of the two target phenotypes, you could have predicted the outcome by using Hebb's rule. Here is the summary, okay? So for the two environments. Some evolve at some activity patterns. Some activity patterns evolve at a constant positive rate. These arise from pairs of traits that are positively correlated in both patterns. For example, genes 2 and 6 are ++ in S1 and -- in S2. Likewise, negative interactions evolve at a constant rate between pairs of traits that have opposite signs in both patterns. And finally, when the correlation of a pair of traits in one pattern is contradicted by the correlation of that pair in the other pattern, for example, in the two target vectors, S1 times S2 is bigger than 0 in environment S1, and S1 times S2 is smaller than 0 in S2, the corresponding regulatory interactions are unable to record the correlation of either target pattern and remain near 0 on average. So this is the explanation for the zeros. This effect is now real memory. How do you know it? You know it because you can do the same thing as you do for the Hopfield network. So you can start from, you can have the evolved B matrix and launch the system from random initial patterns. And they will spontaneously settle either into one pattern or the other target pattern. You don't have a mix between the two. It's like the Hopfield network. And what is also important, that once a system has learned the previous pattern, and now you want to select again for the appearance of that pattern, then it goes much faster than when you were selecting it in for the first place. So the system stores the memory of the previous selection, even if another round of selection of 2,000 generations were superimposed on the previous pattern. The question is, of course, the memory capacity of the system. So how many phenotypes you could store? For the Hopfield network, we have an absolutely accurate number how many different patterns the Hopfield network can store. But there are other versions of recurrent neuronal networks, and for them we know that that depends on many things. And also, for the Hopfield network, there is a very unpleasant feature. If you start loading the Hopfield network with new and new memory patterns, you have a point that's the carrying capacity of the Hopfield network when you have catastrophic forgetting. You have forget everything that you have learned. That's not cool. There are other recurrent neural networks that can do it by palimpsest memory, which means that you can load newer and newer memories, and you will forget the oldest slowly, right? Which makes a lot of sense. I am very curious to see whether these genetic networks that we discussed, evolving in agreement with these pairwise Hebbian rules, whether what's their memory capacity, how many phenotypes you can store, and whether the memory it has got catastrophic forgetting or palimpsest memory or whatever. These things have never been investigated because these investigations are very new. So the point is that the B matrix can store memory patterns. The memory patterns behave like memory patterns in neural networks. They can be recalled. You go either to this, so never a mix of the two, and so on. And that helps you when you encounter the similar situation. Then you can switch to that situation very fast by a little bit of fine-tuning. You still remember how to do it. So these are different experiments. Very, very nicely done. This is a well-known phenotype, right? You will recognize this gentleman, supposed to be Charles Darwin. And this is another target phenotype, that's Donald Hebb. Now, of course, this is completely arbitrary, but since we are dealing with strings, and the string can be cut into pieces, and then you can have activity levels that, give you the pixels of the two images. It's for illustration, it's a very nice thing. So if you train the systems or evolve the systems under two selection regimes, here are the results. So one is increasing noise. That means mutations on G, which means the direct effects of the gene, and the resulting adult phenotypes bottom row. So this is the direct effect on the genes. This is the capital G that is evolving fast. Now, if you introduce many mutations into them, then the direct effect conveys no information whatsoever anymore what pattern you want to have. But the B matrix has it, even without the direct effect. Here you actually don't have any direct effect anymore. And you see for how long you still, if you started with Darwin, you will keep the Darwin. And only when you have a lot of randomness in the direct interactions, then you have what either Hebb or Darwin, but never superposition of both. It behaves just as these neural networks should do. Another one, random G, which means that you randomize. You don't mutate, you completely randomize, but again and again, these are different random patterns for the direct genetic interaction. And here are the resulting adult phenotypes. The resulting adult phenotypes are, again, either Hebb or Darwin, but nothing in between. It has got pattern completion, partial G, top row, and resulting adult phenotypes, bottom row. You see, for a very, very long time, you can actually restore old Donald Hebb. And only if this is very, very partial, then you have a problem, and it becomes random. So it's either Donald Hebb or Charles Darwin that appears. That may be interesting. Of course, it's obvious if you know these dynamical systems, but the system breaks down if the interaction between the genes is linear. It's not filtered through the sigmoid function, right? If it's not filtered through the sigmoid function, you see, then you have got these superpositions of the images. So you don't have a real memory. For real memory, you need the nonlinearity of the system. And its sigmoid function is, of course, kind of a default for genetic regulatory networks. As for the transfer function of neurons, by the way, so they are, again, similar to each other. Now, can the system generalize? That's the next question. Can it generalize or not? And this I'm going to show you with a different example. First of all, I have to say that there is a lot of evidence for modularity in organisms, evolved molecules, living systems. Here is a certain type of, here is an RNA molecule, and it has got a phenotype. The RNA molecule folds back. It's like DNA, but it can fold back onto itself, and that's the phenotype, you know. That's the two-dimensional. It can also, it can form a 3D structure as well, but that you cannot calculate yet. So, life is tough. But okay, so here you see that when you melt you increase the temperature, the different modules open up one after the other. This is an empirical modularity. And here are actually good old Darwin's finches. It was actually the Grant husband and wife who realized that it would be nice to look at Darwin's finches again with the modern tools of biology. You know, Darwin's finches are those creatures that on the different islands have different forms of beak because they have different kinds of nourishment. Beautiful example of natural selection. By the way, when Darwin found them, he didn't even know that they were finches. I mean, just for the record. That was classified properly later, but the phenomenon was very important. And they have found out that the beak varies actually according to certain dimensions, and these dimensions are influenced by various ways, by different genes. So it's like having these, it used to be called these tuners, you know, on these musical instruments. These tuners can be regulated, and by the regulation of the tuners, you can actually form the beak in the three-dimensional rendering. So there is a lot of empirical modularity in biology, and this is probably important. Why? Because modularity is also has a very strong effect on evolvable representations. Here is a nice chair, a nice table, and imagine that you want to breed for the customers tables by a genetic algorithm, right? Now you have to represent tables somehow in the genetic algorithm. And you have to represent the variability of the tables. You can have different options. One option would be, somebody could program it like that the legs of the table would vary independently, right? Others would have the idea, whatever you do, whether it's thicker or longer or whatever, it will always happen to the 4 legs at the same time, right? Obviously, tables that are encoded like this are much more evolvable, because for the other encoding, most of the stuff that you are going to produce by variation will be junk, okay? So representations are extremely important for solving problems by whatever algorithm you want. Maybe it's the representation that is the most important. I remember when they asked Bobby Fischer, how many steps you think ahead in your chess plays? He said, usually it's 1, but it tends to be the right move, right? And they asked him whether he was joking. No, he said, no, I don't think. I have like a kind of a constellation, and I know that out of that, there's the hypothesis is, and this would be a very interesting research for cognitive scientists, the hypothesis that professional chess players have a different representation of chess in their heads from ordinary people. It's a different encoding, and therefore, of course, the neighborhood relationship can be also different, right? Distances can be different in a different angle. Okay, so that would be very... Now, that's just to introduce that representation is extremely important, and you can have good representations and bad representations for evolvability. This is a recognition by Kirchner and Gerhardt, is the idea of facilitated variation. Now we come to an important point that I mentioned in the introduction, that yes, in the genotype space, you can argue that the mutations are random, but very often you find for real organisms that this so-called random variation is channeled through development into phenotype space such that it is more likely than purely random to produce something that is potentially useful. It's like, having the right representation of the table. It's exactly the same kind of thing. So it's if the development of the table is such that the phenotype map will work in such a way that the legs will always have the same length, bingo. And this has been investigated by a theoretical paper, Uri Alom from Weizmann Institute, facilitate variation, how evolution learns from past environments to generalize to new environments. That's cool. And they were trying it basically in their paper on 2 different systems. One was RNA folding, and RNA phenotype as 2-dimensional structure. The other one was logical circuits. And what they found, basically, that there are 2 possible outcomes depending on how you are selecting for a particular performance. If you are selecting just for an end result, for example, on a logical circuits, of course, there are the rules of variation. Okay, what means a mutation here is defined in the system. So if you just look at the outcome of the system, and that's the only criterion how you select, then you will have something which tends to be not decomposable and non-generalizable. However, if you do it in such a way that you are selecting modularly, which means that you figure out from what kind of modules you can produce the outcome that you want. You again will have the outcome, but then it will give you something which you may not have hoped for. That if you are selecting modularly on a composite goal, then this system responds to selection faster if you are selecting for a modular goal which was not presented before. Okay? So indeed, if you are using the modularly varying goals types of selection, you have a more evolvable system than without it. And that was very nicely done by the RNA system with loops and hairpin structures. How come? Why is that so? There must be a mechanism whereby the system quickly switches when you switch the selection regime, right? And they could identify that if you are selecting in a modular way on the RNA phenotypes, then this evolvability is due to 3 trigger nucleotides, because when these 3 trigger nucleotides are just point mutated, the phenotype that you can calculate from energetics radically transforms. Okay? So because of this modular selection, you are co-selecting for the switches that allows you to go for a different composition of modules than when you change the selection regime. Now, of course, if you continue to do the same selection again, you don't change it, that system is going to forget by spontaneous mutation, that's for sure. But it takes time. So this is a demonstration of a difference on a different system that indeed these systems can even generalize. That means that they can evolve things faster if they have undergone this kind of treatment than without it. So this is the space of possibilities, and this is the modularly varying goal language, and this is the rest. And if you know abstract grammar, then it is true that you can make the statement that if a structure belongs to the same grammatical class that you can define, then you are going to be able to generalize to it. If it is outside that grammar, you are not going to be. Then you need doing evolution in the really hard way. I mean, sort of from scratch, right? Then learning is not good for that. But within that class, learning is very nice. Finally, we made the same kind of investigation for ecosystems. Now, this is more exciting. It depends how you look at it. Why? Because when you are selecting on the developmental phenotypes, it's like reinforcement learning, right? You make some kind of activities, and some activities will be preferred and others will be suppressed. But you do it through replication and selection. So the unit of selection is the whole network, right? So it's reinforcement learning on a whole genetic network. Now, many people have been puzzled for a long time whether the orderliness that you find in ecosystems is just, how should I say, a byproduct, which just happens, or whether there was selection on the ecosystem as a whole. Most people would say no. Most of the time, ecosystems are not units of selection. But there seems to be order. So is the order just an additive thing because the individual populations are ordered and somehow the whole image looks neat? Or can you do something, can you explain something more about the phenomenon? And I don't want to go into the technical details, but we analyzed here a Lotka-Volterra competitive ecosystem. It's competitive because the interaction between the different populations is mutually negative. I inhibit you, you inhibit me. Why? Because, for example, we are partially eating the same resource. Question is if the weight or the interaction coefficients, it's again a double language. If the weights are evolvable, in which direction they are going to go? And by now you can imagine one of the questions is whether Hebbian rules have anything to do with what you observe in the evolution of the interaction matrix. How do you do that? Again, you can select for one environment, or you can have one type of environment and another type of environment. The point is that here we have only individual selections in the different species, right? And it's selected when it grows in the particular community. There is no magic about this. The community as whole is not being selected. That's very important. And the experiment for two environments you do in such a way that you are tweaking the carrying capacity in the different models, and you can assume that in the two environments the carrying capacities of the different populations either go up or go down, and the system wants to adapt to this situation by mutation in the interaction weights. What is the pattern that results? I don't want to become very technical, but I want to tell you, here is a similar image that you have seen before. So here, for a particular experiment, you see the resultant changes in the interaction weights. And this is the pattern that you can predict from the Hebbian rule. And again, this system has got what is called ecological memory. It can restore itself from disturbed population densities and so on. Again, it's either or. It either goes in one attractor or in the other attractor. And again, if the populations have been selected under condition A, then under condition B, and then you go back to condition A, again the switch is much faster than before. Why? Because you again have this double memory effect in the interaction weights. The interaction weights partly or also code depending on the initial conditions for one attractor, but also for the other attractor. And this is a nonlinear system, so it's not surprising. This is what is surprising, that, again, I have to emphasize there was no selection on the community as a whole, right? Just as in ordinary Hebbian learning, there is no selection on the network as a whole. It's only local Hebbian. You are only selecting for correlations, but not at the community or the whole network level, because that would be reinforcement learning. I don't fully like this model, I must say, because there are some limitations. There is this total symmetry that I hate. It's, again, the ghost of Hopfield, in a way. It has got some technical reasons. The other one is the normalization, which makes sense, but only partly. What does it mean? Normalization means that you have this matrix of the interactions, and we assume that the sum of the entries have to be the same this way and also that way. What does it mean? That when you decrease the competition with somebody, then you must pay for it with increased competition with somebody else. Now, this has, if you think about resource competition and so on, this has some justification, but not a full justification, right? So the rigid normalization is not good, but as I said, this is real research, and I don't know how this will generalize. For mutualism, it would be trivial to show that Hebbian learning works. Think about it. It's so obvious that we didn't even do it. And here is now the translation table between the mechanistic equivalence between evo, echo, and learning. And here is the ultimate punchline for unsupervised correlation learning. Positive feedback between activation strengths and connection strengths, like neurons that fire together, wire together. Unsupervised correlation learning, like Hebb's rule, and this is the classical Hebb rule. And evolutionary interactions, individual selection only. Positive feedback between ecological densities and connections, or species that occur together, wire together. Direct effect of individual natural selection on interactions, blah, blah, blah. And then you have a mathematical isomorphism. So this is where we stand. As I say, this is genuine research. It might be extremely important in the long run. It might have moderate importance or very small importance, I don't know. But this is why it's research, and we want to find out. And indeed, we find everybody to find out. Because to unify different fields and gain more insight than before, I think is an eminent goal in science in general. And I don't want to explain it to you because I don't have time left. You can read it in certain papers already, but you can ask the following question. This is where I'm Sherlock Holmes, not Dr. I'm Sherlock Holmes. These were parallel developments. So Richard had these ideas about learning in evolution when I had the ideas about evolution in learning. Okay, it's the other way around. Can evolution happen in cognition? What does it mean? It means the following. When you are developing natural language as a child, or when you are working on the theory of general relativity, or when you are working on an invention, or you want to solve an insight problem, so-called insight problem, whether in your brain in real time something that is closely similar to evolution by natural selection takes place or not, despite the fact that neurons themselves don't reproduce. Otherwise, people would have realized it, much earlier, than that's a possibility. Kurz und gut, as the Germans say, so short and well. The answer is yes. There are at least 3 different mechanisms whereby you can implement what we call Darwinian neurodynamics. Whether it's being implemented in the real brain, we don't know. Why? Because nobody was looking at it that way. You are interpreting patterns in your experiments according to certain paradigms. Neurobiology may already be full of evidence for what we are proposing, but since nobody was looking at the data, nobody realized that what they were seeing. And that's another lecture, but it's a flip side of the coin. And then the claim would be that when the brain solves complex problems, then it actually uses the synergy of 2 things: the classical neurobiological mechanisms and the classical evolutionary mechanisms, but together. And that we can show that when you apply the 2 together, and definitely that is a superior algorithm. I survived. Thank you very much.
Question from the audience?
It's okay. I think there might be a typo in one of your equations. And in the very first beginning, they started off with the Bayesian equation and another equation that was analogous to it. And the denominator, I think, should have a mean on it. You're dividing by a function rather than the mean.
Yeah.
Just if you go back, I'll show you what I mean.
Okay, fine.
Yeah.
You understand that.
Thank you very much for your talk. Why is the learning function a sigmoidal function and not a hyperbolic function?
Well, it's not the learning function. Is the activity function of the summed interactions of the genetic regulatory input, and that is because these kinds of inputs have a certain cooperative effect due to things like allostery and these kinds of things. And this allostery is conveniently modeled by a kind of sigmoid curve. That's
But in the experiments we see actually hyperbolic functions. Or the sigmoidal function is so small you can't even see it in the experiment basically.
I think, okay, so the tradition, because we wanted to put in the tradition of the genetic regulatory network literature. The tradition there is to use the sigmoid functions. But, this would work with other functions as well. The point is that it has to be nonlinear.
Yeah.
If it's linear, it will break down.
Yeah, but it's a hyperbolic function.
Okay.
It's also nonlinear. So.
Exactly. So that is robust to that kind of change.
So I am not a biologist, and I'm trying to digest all of that. It's very, very interesting to me. One question I have is the table that you were showing. I love that idea of evolvability. That's a great way to explain it. The idea of the legs all having to change at the same height. So my question to you is, it would seem just intuitive to me that intuitively that those kinds of features might be things that are key to the success of further mutations, let's say, other kinds of things. So would it be the case that those kinds of evolvability traits would show up earlier in the biological record than other kinds of traits?
Oh, that's probably true.
Is it?
That's probably true because the phenotypes have also evolved, right? So facilitated variation at the cellular level must be more common and widespread than facilitated variation, for example, in birds, because birds are recent phenotypes, and there was not so much time for selection to operate and discover these kinds of things than for cells which are around, for more than 3 billion years.
Yeah.
Yes.
So I guess my follow-up then is I keep thinking of machine learning as I'm listening to the stuff that you're looking at. What I'm wondering now is whether what we're seeing is kind of an extension of that in the machine learning space, that the things that we're seeing now are the kind of the earliest kind of evolvability type traits that may kind of shape the landscape down the road.
Okay, I think I understand what you are hinting at, and that is a very interesting remark at the interface of cultural and technological evolution. But since it's also evolution, these evolvability issues are naturally raised or can be raised in that domain as well. I agree with you, but you have, but I also would caution against, how should I say, too much hand waving. So when you want to analyze such a situation, you have to go into the nitty-gritty. That's one thing. The other thing, as Maynard used to say, you have to do the sums, right? Which means that you have to do a working model of it, because otherwise Verbally you can say so many things. When you are forced to make a model out of it, then you often realize, may not be so easy, or you hadn't thought of a certain element. And so it's good to start. There's nothing wrong with starting verbally about such questions, but ultimately you must be able to have an idea how it really works. Yeah, Tom.
First of all, thank you for a wonderful talk. That was really stimulating. I was wondering, and this may be beyond the scope of what you've addressed so far, but you, in some of the models, you deal with mutational noise as being an essential part of the makeup. I'm increasingly interested in developmental noise because we see now growing evidence for the impact of developmental noise. Now, at one level, obviously, if you've got developmental noise, it will simply weaken the opportunity for the kind of the learning within evolution and ecosystems. But I just wonder whether you had or have any thoughts on if you were to incorporate developmental noise within this model, how evolution would act to constrain developmental noise. This was another great interest of Hopfield's in the 1970s, and whether it should be a part of the story.
I agree with you, Tom. This is a very interesting question. I could say I haven't got the foggiest idea, but that's not completely true. But let's do the sums first, and then I don't want to make any pompous statement about it now. But it's a very important question, and of course that's one next step in the research program. So thank you for pointing this out.
This may not be a very professional question, but I still want to ask. So when we train the Hopfield neural network or something like this, the parameters are stored in the nodes. But for a biological system, each generation they just die, and how the information is stored for a specific species.
But the information is stored in the B matrix. Right? The B matrix is the interaction between the genes that you inherit, right? It's stored because you get it from your parents in a way, right? After mutation, possibly recombination, but our model didn't include recombination because for this particular question it didn't make much difference. But you know that you store for your lifetime because you inherit it, unless you have a somatic mutation or developmental noise is masking the inherited value. But it's basically inherited. In the model, within one generation, it's fixed. Right? It changes when you get to the next generation.
So Eörs, can you look at the brain's development as a development of composite systems, composite Hopfield networks? Say, for instance, during the evolution of the human brain in the animal kingdom, you had different compartments, different networks, including motor network, including memory networks, including sensory networks, and so on. And these can be regarded as separated compartments of a larger brain. And you can regard their development as individual developments, but also there is an overarching, larger component which drives the whole development and synergizes the development of these different individual silos into one brain development.
Yeah, you know that the issue of modularity is a long and treacherous book in neuroscience. You can understand it either structurally or functionally. Right? It's easy to identify some of the structural modules at least. The question is, what is the functional correlate of a structural modularity? And even though it's not absolute, but given a certain developmental pathway, it is true that some parts of the brain are the convenient places during development for the final localization of a capacity. So as far as I know, when, and that's another field of interest for mine, if we are looking at the origin, I mean the emergence of language, linguistic capacity, what happens is that when in very young children the activity is much wider, also for syntactic tasks than the Broca area. But in most healthy individuals, finally it gets localized into what we call the Broca area, because what I think is, given the contingencies of the other connectivities, that's the best and most functional place where it gets localized. Yeah? But you get it. So I think that this localization phenomenon is also a dynamical process. It's like people trying to take the most comfortable chair in an home. Now, if you have a disturbance, then we know that the plasticity can be enormous, right? If your favorite seats are taken, then you will look for another one, okay? And you can still enjoy the lecture to a great deal and so on. But that's of course dependent on when you have the disturbance, because you have critical periods and these kinds of things which are making the whole stuff complicated. So I think that this modular aspect of the brain is real, but I think it's more subtle and more dynamic than you tend to read about it, even now in textbooks, which is surprising.
Any more questions?
Thank you very much. Is this on? Yeah? Yeah. I'm really taken by the breadth of all of these things that are analogous, population dynamics or ecological dynamics, brain functions. I'd like to add, and you even had electronic circuitry in there, which is one of my real interests. And I would like to, this is a comment, but I have two quick questions for you. As another comment, I would say an awful lot of economics works this way. You can think that, say, a stock market, for example, is an ecology of predictors. Each person's thinking this might happen, that might happen. Given the current situation, I'm expecting or forecasting this to happen. So you have mutual ecology that's always changing of predictions. You can have ecologies of strategies. I'm starting up a company, say, in Silicon Valley. Somebody down the street is starting. And so you get very rapid mutual adaptation and the emergence of ecologies of strategies or that eventually congeal into companies, etc. So this recurs in economics, and economists don't write about things much this way, but I think you'll be ahead of the game if you widen this into what's called non-equilibrium economics or complexity economics. Two quick questions, or love to get your comments on. I was sitting here thinking that adaptation, as it's interpreted, say, by Fisher, or even by Darwin before that, tends to think in terms of one species, say like the finches and the Galapagos, one species adapting slowly by the accumulation of small differences, as Darwin pointed out. And it seems to me, you could summarize what you're saying as you're extending that idea of a single species adapting to an entire ecosystem adapting and mutually learning and possibly reaching a stable state where it's then frozen. But in other words, you're extending adaptation and learning from just one species into a whole ecology and showing how that echoes across many fields. The other question I have is, you said quite a bit about modularity, and that's very interesting. Maybe I didn't quite get all that you said, but my question is, you may have covered this, but is modularity something that emerges? I think you were talking about if there are given modules here, here's what might happen. But is there a tendency for an ecology to coagulate or coalesce into interacting modules, and some of those interacting modules to coalesce into higher level modules? So, anyway, any comments?
Thank you. Thank you very much. I greatly appreciate them. Application to economics. You have already suggested this to me by your sheer name. Let me tell you how it happened. So before we came here, I was glancing through the book, who was going to be present. And I said, okay, Brian Arthur again. Ah, lovely. And then I said, okay, yeah, but... and then I had, my association cortex must have... So I thought of that. But of course, I didn't do any work yet, in the economic... But it was your name that triggered me to think that. Thank you very much for the encouragement, because that is something that we need, right? We are miserable humans. The second thing is that indeed this view of ecosystems, that you have to take it really with a grain of salt, what I'm saying here, because I'm not really happy about some of the detailed modeling assumptions that we have, but I have a hope that it can generalize still. We shall see. But suppose it can generalize, then the answer is yes. It really widens the notion of adaptation in biological populations. I absolutely agree. Because of this implant of the Hebbian mechanism into something which was not seen clearly before. The third question was modularity, but what was it about? Sorry.
Modularity emerge endogenously.
Oh yeah, modularity emerges endogenously for several reasons. One is the division of labor. Right? As we know, if you look at division of labor in biological evolution, right? Specialized organelles in cells and so on. The situation that you have is not unlike what Adam Smith described in The Wealth of Nations. So, and that already showed, because these functions can and do disturb each other, which means that a jack of all trades is a master of none, quite often in biological and physiological functions as well. So if the extent of the market is large enough, there is a size issue as well, then of course it pays you to have specialized things, and specialized things will, by definition, have, they will act as different modules, because they will have a limited subset of interactions, quite close interactions inside, and only some interactions to the other. Yeah, that's one of the main reasons why modularity.
Well, you see it in your models.
Oh, yeah, many models we saw that. Not only mine, but but it's a valid point that modularity does. The other thing is the cost of complexity, as Günther Wagner calls it. If you want to change, if you don't have modularity, then the pleiotropy would be extremely widespread, which means you change one gene and you change everything. Now that's not a good thing to do, because most of the time you will just screw up the whole organism. So there is selection for packaging in it a way that is now consonant with the facilitated variation also. And these hang together, these considerations. Now, they didn't hang together 20 years ago.
Thanks again, Dr. Harsh. We would like to present you a token of
How
appreciation.
surprising. I think we should go up there.